Okay, let's get started. I'm happy today to introduce Regina Fairbanks, who is a third-year Pop Bio grad student working on maize and human coevolution in the lab. Regina came to the lab with a strong background in archaeobotany, having worked everywhere from 19th century archaeological remains, everywhere from 19th century South Carolina to ancient maritime Greece. They're also super interdisciplinary. So in previous work, they've worked on phylogenetics of ginseng to chromatin dynamics in Drosophila, and everything from microscopic analysis of archaeological remains to the population genetics that you'll hear about today. Regina is also deeply invested in science communication, from public seminars to developing museum displays to helping the lab develop an educational video game. So if you're interested in SciComm, I would definitely, and volunteer work on letters to a pre-scientist. So if you're interested in any of the range of science communication stuff, I'd encourage you to talk to them. Yeah, okay. With that, I will turn it over to Regina to talk about their work on maize domestication. Please, go for it.
Thank you. If I'm not loud enough, just wave your hands at me in the back. Sometimes I'm a bit quiet. So thank you all for coming to hear about my ongoing and planned research. But before I get into that, slides. Okay. I just want to provide some context for how I ended up here working on domestication in maize. So in general, my overarching interest is in the relationships between people and plants, especially how those relationships have changed over time and how those relationships shape the plant and people partners. Like a lot of us who are interested in plants, we develop these interests from really personal experiences, things like encountering plants in the garden, natural environments, and for some of us, especially in the kitchen. I'm from the Philly area, which likes to call itself America's garden capital, as it's home to a few dozen botanical gardens, including the world famous Longwood Gardens and America's first botanical garden. So I visited these a lot growing up because my mom is a home gardener, and we also visited nature preserves where we would see some of my favorite plants like the skunk cabbage. But for me, I'm really interested in plants and their contributions to food, especially because I think they help me connect to my heritage. My grandfather's family had a store in Philly's Italian market where they sold Italian produce and other goods. But noticeably absent in my personal journey to plants is maize, which is the plant that I've spent a lot of my time thinking about. And I've come to appreciate it as an academic researcher, but I want to take some time to acknowledge the cultural and spiritual importance of maize to many indigenous groups throughout the Americas, both today and over the past thousands of years. Many of these people literally think of humanity as being made of maize, and it's their ongoing care and stewardship that's produced the diversity of maize that we see today. So to start off, around 10,000 years ago, in many different places across the globe, people and plants began forming a new complex relationship, which is familiar to many of us as domestication. And we can think about domestication as this process where wild plant species encounter new environments and selective pressures under human cultivation, which ultimately leads them to evolve distinct morphologies from their wild relatives. Often they're doing things like evolving traits that make them better adapted to disturbed environments, like larger seed sizes, or adapting to human-mediated reproduction, like non-shattering ears. And a lot of us like to think about domestication as a special case of plant-animal coevolution, where on the one hand, you have humans controlling the fitness of the plant by controlling its reproduction, and on the other hand, the plant shapes human fitness by providing often a stable, abundant food source. So over thousands of years, we've seen that hundreds of plants have undergone domestication, and because domestication gave rise to agriculture, it has far-reaching consequences not only for the plants and people partners, but also for the entire planet. So domestication clearly is very interesting to anthropologists and archaeologists who think a lot about the origins of crops and the origins of agriculture, but it's also been of interest to evolutionary biologists for the past 160 years. As Darwin began to develop his evolutionary theory, he was thinking a lot about domesticated species, things like variation between different breeds and the role of human selection in shaping these domesticated species. And this interest continues today, where crops like maize often provide tractable models for studying evolutionary processes, especially because a lot of interest in these crops for breeding efforts in agriculture means that there's a lot of resources funneled into them, and we can use those resources to study evolution. So when we're thinking about what we know about domestication and where we want to go, early domestication research was really focused on a couple questions, things like where particular crops first appeared and what their wild relatives were. And people were focusing on things like Pavlovian centers of diversity and cradles of agriculture. And later, with improved archaeological dating methods, people also began to understand when domesticated crops showed up in the archaeological record. So we now have a pretty decent idea about where and when many domesticated crops first arose. But what we're still trying to understand is how and why domestication happens and what the process of domestication looks like for the relationships between people and plants over time. So I'm working on answering some of these open questions in maize, which is not only one of the most dramatic examples of domestication, but also one of the most well-studied. Throughout this talk, I'm going to be mentioning teosinte, which refers to the closest wild relatives of maize. And maize began to diverge from teosinte about 10,000 years ago, when people began to manage some ancestral population of Parviglumis teosinte, a specific subspecies. So the noticeable differences that you see during this dramatic process was that, on the one hand, teosinte produces many dozen small, tiny ears. Each of these only have a handful of kernels, and all of those kernels have this really tough fruit case that's inedible and makes them look like tiny pebbles. But during domestication, what we see now is one or a few ears per plant with many kernels, and all of these kernels are fully exposed on the ear beneath the husk. And most of these adaptations facilitated things like harvesting and processing of the grain. So in the thousands of years following domestication, people moved maize throughout the Americas, where it encountered new environments and cultures and adapted and diversified. And what happened in many regions is that maize became an important part of the food production systems, and it took on really important cultural roles. But we still don't fully understand why and how maize domestication occurred and how it subsequently evolved in tandem with different human populations. Now, for my research, I'm doing two major projects. The first one is that I'm looking to understand the origin of the genetic variation that underlies a key domestication trait in maize. And for the second, I'm exploring the relationships between human and maize population sizes over time. So before I mentioned that we now have a pretty good idea of when domestication seems to have occurred for many crop species, but what we don't understand still is when specific domestication traits and the underlying genetic variation first appeared. So whether domestication traits and their alleles existed before domestication started or after selection had already started has important consequences for understanding how quickly domestication might have occurred. And this question is also of interest to evolutionary biologists more broadly, as case studies in both natural and experimental populations have shown that pre-existing or standing genetic variation can play an important role in adaptation. But the relative importance of new versus pre-existing genetic variation remains debated. So I mentioned that we're interested in distinguishing between two different scenarios for the origin of genetic variation underlying domestication traits. So in the first, genetic variation appears during domestication once selection by people has already started, and this is the de novo model. And under a de novo model, you don't see domestication traits in the wild or in the ancestral population, and you see them only appear in cultivated populations. And the other scenario, this genetic variation does exist in the ancestral populations. It's segregating, often as neutral variation in the wild. And then during domestication, selection for that trait leads it to increase in frequency in the population from what was originally standing variation. But for addressing this question in maize, a major limitation is the poor archaeological record before and during domestication. So surprisingly, people have not found any evidence for teosinte in the archaeological record before we start seeing maize. So we don't know what that teosinte looked like or what traits might have been segregating sometime in the past. And we also have a long gap during the actual domestication process. So the earliest evidence for maize that we have are these microfossils around 9,000 years ago. And these are little silica structures that can tell us that maize was there, but it can't really tell us a lot about what that maize looked like at that time. We don't really know what those maize ears looked like until 2,000 years later, when the first archaeological ear appears. And at this point, it already has most of the traits that we think of as domestication traits. So we can't track the traits directly in the archaeological record, but what we can do now with population genomic data sets is look at how the genetic variation has evolved over time. So this is an important question, but it can be hard to answer. You need to know which spots in the genome were under selection and which variant at those sites contributes to the maize-like phenotype. But for maize, there are a handful of well-studied domestication loci where we both know where those spots are and which variant contributes to the maize phenotype. So people can go out and look for those variants in modern teosinte populations. And so we see for these handful of well-studied loci, most of them involve selection on standing genetic variation, where people have found the maize allele in the wild. So some important examples are the non-shattering ears and this change in plant architecture, which do segregate in the wild today. So it seems that pre-existing or standing genetic variation might have played an important role in maize domestication. But there's one major exception that people have talked about, and this is TGA1, teosinte gloom architecture. And this is considered to be maybe the most important gene during maize domestication. And what we know is that a single mutation in TGA1 helps with a critical step in maize domestication, which is the loss of this fruitcase that I mentioned earlier. So as a reminder, the teosinte relatives all have these really tough and edible fruitcases, which are thought to protect the kernel from seed predators by allowing them to pass through the GI system of things like birds. But it makes them pretty terrible grain crops because it's really hard to remove and use the kernel for food. So during domestication, people presumably were, one, protecting their population of plants from seed predators, and two, they might have been selecting for exposed kernels to increase access to the grain. And we know that TGA1 played an important role in this, as the region in the genome that has TGA1 seems to explain up to 60% of the phenotypic variation in kernel exposure and related traits. And people have thought that TGA1 has a de novo origin for at least a few decades now. Beginning in the 1970s, there was a field survey of wild teosinte. They were explicitly looking for maize-like traits in the wild, trying to answer this question of de novo versus standing genetic variation. And they didn't find any evidence that exposed kernels were standing genetic variation before domestication. And they had this idea that exposed kernels would have just been too deleterious to persist in the wild for more than one or a few generations. And then later population genetic studies, which were sequencing teosinte individuals at TGA1, seemed to agree with this, as they were only finding the teosinte allele and not the maize allele at that site. And we also see these microfossils that I mentioned earlier. They tell us a hint that TGA1, the maize TGA1 allele was already present and at high frequencies in early populations. And then by the time that we have ancient genomes, they also have the maize allele of TGA1. So it seems that the de novo appearance of TGA1 was this like eureka moment that people talk about in maize domestication, where the process only really started after this allele and the trait appeared. So both field surveys and previous population genetic studies are suggesting that there's a de novo origin, but we have some population genetic evidence that this might not be such a simple story. So with the arrival of large genomic data sets, people have been conducting genome-wide surveys for selection in maize, looking for sites in the genome that underwent selection during domestication. And these scans are usually well-powered to find spots in the genome where selection occurred, where strong selection occurred on de novo variants. So they should pick up TGA1 really readily, but they often fail to identify it in these scans, which is confusing if it has a de novo origin. So this suggested to us that maybe it's time to consider a standing genetic variation origin for TGA1, where perhaps this allele existed in the ancestral population and just hasn't been detected yet. So we were trying to answer a few questions to get at this. So one, do we find the allele in teosinte when we use larger data sets? Do we find signatures of selection that look like standing genetic variation? Because often the signatures of selection on de novo versus standing genetic variants look different from one another. And also, do we find that the age of that mutation that's important, is it older than when we think domestication started? So like I mentioned, previous studies didn't find the maize allele of TGA1 in wild teosinte, but we revisited this question with a much larger data set of individuals, so up to 130 teosinte individuals, and we do find a few that are heterozygous at this site, so they have one copy of the maize allele and one copy of the teosinte allele. And this is the first evidence that maize TGA1 can be found in wild teosinte populations. The weird thing, though, is that we found this in not the closest wild relative of maize parviglumis, which contribute the most to maize domestication, but instead we found it in Mexicana, which played a role in maize evolution, but only a few thousand years after domestication started. So it's slightly odd that we can't find it in parviglumis, where we might think that it arose in an ancestral population of maize and parviglumis and persists until present day. But we think that future efforts might find the allele in these populations in the future, but it's just that sampling strategies so far haven't been able to detect it. Things like spatial variation in the frequency of the allele could contribute to that. For example, it might be at low frequencies in some populations or missing in some populations and can be found in other populations. And we know this is the case for other domestication loci. For example, TB1, the maize allele is absent in some populations, but is at relatively high frequencies, like 44% in other populations. But there is a small caveat to this suggestion in that the presence of TGA1 maize allele in modern teosinte doesn't conclusively prove that it originated in the ancestral population and has been hanging around for the next 10,000 years until present day. Because this pattern could also arise from post-domestication gene flow from maize back into teosinte. And we know that there has been and continues to be ongoing gene flow between maize and teosinte. But we can't rule out gene flow with the things that we can look at, but we can rule out recent gene flow. And when we looked at the length of that maize-like chunk near TGA1, it looks like if gene flow is responsible for this, it's at least dozens or hundreds of centuries ago, which suggests that it could hang around as standing genetic variation, which previously people argued it was just too deleterious to survive for very long at all. So the next thing we did with this idea that a standing genetic variation origin was at least plausible is that we looked at whether the diversity patterns near TGA1 could be produced by selection on standing genetic variation. So I'm just going to quickly explain how these haplotype diversity patterns can differ between selection on de novo versus selection on standing genetic variation. So first we have this pre-domestication example where we have these individual haplotypes and the white circles represent neutral mutations. So once domestication started and selection begins, in the de novo model, we have a brand new mutation that appears, and this is the orange circle. For the standing variation model, an allele that was previously in the population now becomes beneficial and is selected for. And the key difference here is that the de novo mutation is just on one genetic background or haplotype, and the standing genetic variation mutation is on multiple haplotypes or genetic backgrounds at the time that selection starts. So we see that after the mutation has had time to speed up to fixation, this can result in different diversity patterns, where in the de novo model, we see just one common haplotype in the population, and in the standing genetic variation model, we see multiple common haplotypes in the population that are associated with a selected mutation. So to see which pattern we can observe in modern maize near TGA1, we looked at a data set of around 500 modern maize individuals, which is represented here. Each strip is an individual haplotype, and mutations are plotted along the x-axis as these teal ticks. And I've highlighted the causative mutation as this asterisk, and this is fixed in maize and it's thought to be that single mutation that contributes to this exposed kernel phenotype. And what we find is that it looks like there's more than one common haplotype near TGA1 in modern maize, but there's at least two common haplotypes, which seems maybe more consistent with a standing genetic variation origin for this mutation. But there are some other factors that can result in this pattern of multiple haplotypes. So we ran some population genetic simulations to see what could give rise to the patterns that we're seeing in modern maize. And I did this just with a couple demographic models that have been inferred for maize, and these capture some of the features that we think are important for maize during domestication. So first, I simulated an ancestral teosinte population to generate neutral standing genetic variation. And then we have this population bottleneck, which is a common feature in domestication of many crops. And the things that differ between these ones is the more simple model has an instantaneous population bottleneck, and the other has a more protracted population bottleneck that occurs over thousands of years. And then we have the beginning of domestication at around 15,000 years ago. And this is when we think selection on TGA1 could have started at the earliest. So in the de novo simulations, I introduced a beneficial mutation at this generation. And then for the standing variation simulations, I took an allele that was previously in the population and just changed its selection coefficient to some positive value. So it shifted from neutral to beneficial. And then after that, we know that there's been really substantial population size growth in maize since domestication. So that was captured in these models as well. And then I don't have a nice visualization of it, so I'll just tell you verbally that we find for both of these demographic models, the standing genetic variation origin produces haplotype diversity statistics like those that we see in modern maize at TGA1. But the caveat is that under the simple population bottleneck model, the de novo origin doesn't produce these patterns, but it does under the complex population bottleneck model. So that was suggesting to us that a standing genetic variation origin can produce patterns that we see in modern maize, but it's not required to produce those patterns that we see in modern maize. But what we had is some additional information from these simulations to distinguish between the de novo and standing genetic variation origins. So we have the ages of nearby intermediate frequency mutations. We have these from the simulations. And then we also have estimated ages from the modern maize genomic data. So under a de novo origin, most of the nearby mutations are going to be pretty young. They're going to be younger than the de novo mutation itself. And then for the standing genetic variation origins, there will be more old nearby mutations in the sample. So when I look at these from the simulations, what I have on the y-axis is the log 10 generation in the past, and then I'm going to show the de novo first. And what I have in gray are the mutations that are younger than the selected mutation, and what I have in the different colors are mutations that are older than the selected mutation. And I have the two different demographic models in teal and orange. So we can see that this meets our expectations that de novo selection on de novo mutations will produce a lot of young nearby mutations. And standing genetic variation models will produce more of these older nearby mutations. And when we compare this to what we see in our sample for modern maize, is that they resemble more closely the standing genetic variation models. They aren't dominated by a bunch of young mutations, but instead have some young, but mostly old mutations nearby. And because we were estimating the ages of mutations near TGA1, what we did was estimate the age of the causative mutation itself in TGA1 to see whether it had occurred before domestication started. And what we find is an age of around 23,000 to 33,000 years ago. And this is much earlier than even conservatively early estimates for when maize domestication began. So we don't have a precise day like when maize domestication started, but we do have some ideas of around when it probably occurred. So the earliest estimates come from some population genetic estimates of when modern maize diverged from teosinte populations, and this is around 15,000 years ago at the earliest. And then we also have an idea from the archaeological record for those microfossils I mentioned earlier, where it seems like domestication was already underway by 9,000 years ago. But in either case, our estimate for the age of TJ1 is around 10,000 years or more older than when domestication started. So in this case, it seems like TJ1 probably arose in an ancestral population of teosinte before domestication started. So starting to conclude, this idea about TJ1 is that previously people really thought that the de novo mutation in TJ1 was this event that started maize domestication. But we're suggesting instead that TJ1 seems to be like other domestication alleles in that mutations previously existed in wild unmanaged teosinte populations, and the start of domestication didn't rely on the appearance of new mutations, but instead was just like reshuffling variation that already existed. It's kind of hard to overstate if you haven't read the maize literature how important people thought this de novo origin for TJ1 was. But a lot of that was based on a belief that exposed kernels and TJ1 were too deleterious to survive in the wild, but people haven't been able to actually test the fitness effects of TJ1 and exposed kernels in the wild. So we're suggesting that in the future, it might be nice if we can take some of these domestication traits and alleles and see how they affect the fitness of wild relatives. And we should also rethink assumptions about what selection was like before and during domestication. So we spent some time thinking about the origins of TJ1, which is one really important gene. We also wanted to think about some genome-wide patterns for the relative importance of de novo versus standing genetic variation. So we're still working on this analysis, but what we've done is one of those genome-wide scans for signatures of selection, like I mentioned earlier. So we find some spots in the genome that we think have undergone selection in maize, and then we calculate this haplotype diversity statistic at those spots, and then compare them to simulated values. So I have plotted on the y-axis this haplotype diversity statistic H2H1. And we find that under the de novo models, there's pretty low haplotype diversity, and then under the standing genetic variation models, there's increased haplotype diversity. And then when we compare that to genome-wide data for maize, we see that a lot of the haplotype diversity patterns can be better explained by selection on standing genetic variation as opposed to selection on de novo mutations. So beyond that handful of well-studied domestication loci, it seems like standing genetic variation could have played a bigger role in maize domestication. So for my first project, I was focusing in on domestication itself. And for my second major project, I'm going to be thinking about the relationships between humans and maize since domestication over the last thousands of years until European colonization. And I'm going to be doing this by exploring the relationships between human and maize population sizes. So I mentioned earlier that the relationship between people and crops, including people and maize, is a coevolutionary relationship where interactions between humans and maize can reciprocally affect the evolution of each other. So we know that from the archaeological evidence, there is a strong connection between human and maize population dynamics, where in many places, archaeological data suggests that maize allows for human population expansion and the development of large settlements. And from the maize perspective, like I've mentioned, there's a lot of adaptations that make maize easier to harvest for people and easier to process for people. But what that does is eliminate its natural seed dispersal abilities so that maize now relies entirely on people for reproduction. The premise behind this project is that those coevolutionary relationships should be captured in correlated population size changes. So far, we don't have a good idea of what the relationships are between those population sizes over time. But what we do know is that there's a lot of variation in the timing of when maize arrives in a region and how quickly it's adopted as a staple between and within regions. So we know that maize kind of moved relatively quickly into South America after domestication, even before key domestication traits were fixed. And it took surprisingly more time for it to move into the Mexican highlands than it did for it to move south. We also know that it arrived in the American Southwest by around 4,000 years ago, but then it took a couple more thousand years to move east to Central and Eastern North America. And we also see there's gaps that sometimes are thousands of years long between when maize arrives in the region and when it becomes a staple. So this happened in the Maya region and in coastal Peru, but in other places like the American Southwest, we see that maize arrives and then very rapidly becomes a staple. So with that said, there's variation in the timing, but we think that the relationship between the human and maize population sizes should be similar. And yeah. So to think about this question, what I'll first be doing is inferring the demographic histories of maize populations in Mesoamerica and Western South America. I'll be doing this in conjunction with some of our collaborators who will be inferring the demographic histories of human populations that are associated with these maize populations. And then we're going to compare those patterns of population size change with existing archaeological data for things like intensification of maize use and some proxies for human population size. So for the demographic inferences, our collaborators in Mexico have a large genomic data set of indigenous people, both in Mesoamerica and Western South America. And then for my maize demographic inferences, I'll be using a published data set of traditional maize varieties from Mexico that was recently published, as well as some older whole genome sequence data from traditional maize varieties in Western South America. So just for what this might look like, I have a really simplified hypothesized example. So the period of interest is from 500 years ago up to maybe 9,000 years ago. When you translate this to human generation times, that's about 20 human generations ago up to 300 generations ago. So in this example, we might expect a large increase in human population size when maize arrives in a region and becomes a staple. And for the maize, this is an annual seed crop, 1 generation per year. So this will be about 500 generations ago up to 9,000 generations ago. And what we might expect is small population sizes when maize first appears in the region, and then a large increase in population size when it becomes a staple crop for the people in that area. This might be because when it's first introduced, there aren't the agricultural and culinary practices that are needed to really intensely use maize, but over time those are developed and maize can become a staple. And from these demographic inferences, what we would like to do is look to see if there's correlated population size change over time. And then we'd also like to compare them to archaeological evidence, like I said, for things like technological developments that allow for better maize processing, things like increase in maize use and consumption that you can tell from isotope data, and other kinds of archaeological information that we already have, especially for these two well-studied regions. And then the other benefit is that we're looking at 2 different regions. So we'd like to see if there are similarities or differences in these patterns across space and between different cultures. I'm going to wrap up a little bit early. So for some acknowledgements, thank you to my QE committee, also some funding sources. I'd also like to thank the 3rd year cohort and other people from Popeye Grad Group, and the Eve staff, who's great, and also Josh. Yeah, and lastly, like I mentioned before, about 10,000 years of indigenous farmers have created this diversity of maize, which is probably the most diverse crop and is really beautiful. And yeah, just thank you to those people. Any questions? Yeah.
Regina, that was a really great talk. I wanted some clarity on the maize teosinte timeline, and maybe you may have said this already, but was there pop divergence of maize and teosinte, and then that ancient maize was domesticated, or was the domestication of teosinte that created maize?
Yeah, I think the latter of what you're saying is that there was there was wild teosinte, and people started to manage some group of that wild teosinte, and then that eventually evolved into maize. Does that answer your question?
Yeah.
So I have a question really broadly in the space you're working in. Do most crops have a single origin, or are there species that have been domesticated with more or less the same outcome multiple times?
Yeah, there are some that have been domesticated multiple times. For maize, it was just once. Yeah, but I don't have an idea of like more, yeah.
So sorry, how do we know that? That it was domesticated just once.
I think mostly it's like some phylogenetic relationships. Yeah. That's all I can think of.
Thanks. Fun to think about.
Is there good evidence of any kind of a role for hybridization at some point along the way, being an important sort of back crossing with teosinte? And if so, what's the sort of, the most recent sort of example of that?
Yeah, the big thing that people talk about and that a lot of people in the lab work on is hybridization with that other subspecies, Mexicana, which probably contribute to maize adaptation to highland environments. And actually, the hybridization of that maize with Mexicana replaced all the other maize throughout the Americas. I don't have a good idea of what the most recent, like, there's still ongoing hybridization in the regions where maize is grown, where teosinte still survives. Yeah.
There are examples of, talk about people thinking that TJ1 was sort of this key domestication gene for maize. Can you think of examples where there is one that is a de novo key domestication gene for some other crop? I can't, so.
I mean, maybe non-shattering in rice, just because a lot of the domestication genes in rice seem to be from de novo, but I'm not sure.
Okay, so no more questions. Thank you, Regina.